Hi everyone. So in this video we're going to cover, Tim and I are going to cover experiment 2. And I wanted to remind you about the outlines, and these are available in the lab manual, but these colored outlines are available in the description below. And the red stuff here is the stuff for experiment 2 that you should do before coming to lab. The green stuff you're going to do during lab, and you could kind of have an Excel file here, or you could kind of just write this down because there aren't as many calculations or calibration curves or statistics like you had to do in experiment 1. So we leave that choice to you guys of how you want to do this. And then the conclusion part, which will be due at the end of your three experiments, and then your whole lab report for the first 3 experiments, so you can save the blue part until the end. Now, I just want to remind you one more time of why we're doing these outlines. These outlines are super important because they help you prepare for the lab, and at the same time, they give us an opportunity to give you feedback on your experiment before your lab report, which is worth a bunch of points, is actually due. So, said simply, the more time you spend writing a good outline, the less time you'll spend writing a lab report, the better feedback that you're going to get, and ultimately the better lab report you're going to have. So it's really in your interest to spend some time on this. One other tip that I would like to give you is that if I were doing this, I would read through this outline, specifically the red parts of this outline, before watching the rest of this video, just so you have an idea of what to watch out for. Because Tim and I are going to talk about almost everything, if not everything, that is highlighted here. So it's a good idea to have an idea of what we're looking for, and then if you hear us say it, just stop the video and type it. And it's that simple. Or write it down on a piece of paper and type it later, whatever works for you. But you can really make your life easier and more efficient if you're prepared for what you need to know, and then you spend a few minutes. So that's enough about that. I would like to get into the nitty-gritty details, I guess, of this absorbance experiment. So at the end of the day, and we'll talk about this a little bit more later, you're going to be making red dye number 40, food coloring solutions to match an unknown sample that's in the lab. And essentially, red dye number 40 is the stuff that makes red Kool-Aid red or red Gatorade red. It's a food coloring, and it's relatively non-toxic, and it's in very low concentrations. And we're asking you to make a solution that is exactly the same concentration as an unknown or a sample solution that we're going to provide you with. So that's basically your goal for this lab. And there's a little bit more to it than that, and we're going to talk our way through what's a little bit more to it as we go through this video, but I just want to give you a basic idea. Now, there's some fundamental things that you've actually learned before, mostly in general chemistry, and you'll learn a little bit more about in quantitative analysis that I just want to remind you about. And these are some of the theoretical aspects of why things absorb light. So I want to talk for a few minutes about the theory of what's going on. I just want to point out again, and we kind of touched on this in the first video, that the things you're going to learn in this course are things that chemists commonly do. Whether they're working in QA, QC type stuff, whether they're working on research projects in graduate school or for some company or whatever, these are just basic stuff that chemists have to be able to do. And UV vis spectroscopy is a very common instrument, one, because it's relatively inexpensive and stuff like this. So knowing the basics of how it works, really important. If you had a job interview at a QA QC company or for graduate school or whatever, it's entirely possible someone could ask you how this works. Might as well learn it now. You're going to see this again throughout your time here and beyond. So spend a few minutes, watch the video, make sure you understand the fundamentals of how it works. Well, the first thing we need to talk about when we talk about UV vis spectrophotometry is, why do molecules absorb light in the first place? And you actually learned this in general chemistry. So you remember the Bohr's model of an atom with n equals 1, n equals 2, n equals 3, and these ground state electron configurations where you learn 1s2, 2s2, 2p6, this kind of stuff. Well, it turns out that molecules and are somewhat more complicated, and this is basically what's going on here. So what you have is, you have these energy levels of electrons in molecules, and also true in atoms, and you also have these vibrational energy states. And it turns out that these transitions are quantized. So if you have an electron down here and it absorbs a photon with the correct amount of energy, which is of course related to its frequency and its wavelength, energy, frequency, and wavelength are all related by constants, namely Planck's constant and the speed of light in a vacuum, then if this electron gets the exact right amount of energy, it can jump to a higher energy level, and it can jump from different vibrational states. It can also jump from here to, say, here. And these transitions are so-called quantized, which means that they happen with very specific amounts of energy, or said another way, photons with very specific wavelengths. So if electrons absorb photons, then they're going to absorb that light, and that is what we're looking at when we're looking at absorbance spectroscopy. And absorbance is related to concentration, because if a solution is more concentrated and it has more molecules that can absorb light, that solution will absorb more light. Now, if it's a colored solution, like visible light colored solution, you're going to see that based on the darkness of the solution. If you have a very dilute red food coloring solution, it might look like a pale red. If you have a very concentrated red food coloring solution, it might look like cough syrup, which is a really thick, syrupy kind of color because it's got a lot of food dye in it. So this is basically what is happening here. So we have these quantized energy transitions. And it may seem like quantized is this like foreign concept that doesn't exist in everyday life, but you can think about this in terms of like the difference between an escalator and a stairs. If you go on stairs, your elevation is quantized. Every 6 inches on the stairs, if the stairs are 6 inches apart, are a place that your elevation could be, because you could step on the bottom stair or the next stair or the next stair or the next stair. But your elevation is quantized, in this case, by 6 inches. If you go on an escalator, however, and you had like a stop button, and you could stop yourself at literally any elevation between the bottom of the escalator and the top. So stairs are kind of quantized and an escalator isn't. So this is not a foreign concept. So molecules absorb photons of light and an energy transition occurs. Now they can come back down by emitting a photon of light if they're fluorescent molecules, or you can have thermal, it collides into another molecule and this falls down and that energy is lost as a thermal collision or something like this. So there are different ways that it could get back down, and one of them is an emission, but we're not going to talk about that right now. So an absorption is an electron at a lower energy level moving to a higher energy level after absorbing a photon of light. So now what we need to talk about here is the color of light absorbed by the molecule, and I'm looking down here. So in any room or outside or whatever, you're essentially being hit by white light, okay? Whether it's white light, I'm sitting in a room with fluorescent lights, so whether it's the white light from the fluorescent lights or the relatively white light from the sun, this is white light. And you have to remember that white light is all the colors of visible light. If you think of that Pink Floyd album where they put the white light through the prism and it's broken up in all the different colors, so or a rainbow or something like that, the white light is all the colors of light. Well, if you have a red food coloring solution, the red food coloring solution is actually absorbing green light. And you may not remember this, but green light is somewhere around 520 nanometers in wavelength. So it's absorbing green light. Well, you might ask yourself, why is that? Many people think that red solutions are absorbing red light, but it's not the case. They're actually absorbing the complementary color of red light, which is green light. Why? Well, think about it a little bit differently. White light, which is missing green light, the complementary color, appears red. So it's absorbing out the green light from the white light, all the colors of light, and when that green light is missing, then it appears red. So red dye number 40 is this large conjugated organic molecule. And you'll look at, if you look at the conjugation, so double, single, double, single, double, single, double, single, double, single, so on and so forth, the conjugation in this molecule, a lot of conjugation brings these molecules down to be able to absorb relatively low energy light, which is visible light. Many organic molecules with double bonds absorb UV light, but molecules with lots of conjugated double bonds, like this one, absorb visible light. In this case, red dye number 40, as we just talked about, absorbs green light. A couple other things to notice. You'll notice that this molecule is ionic. So it has these two ionic areas. Why is that important? Well, food coloring is soluble in water. If this was not an ionic compound, it has a relatively large number of carbons and relatively few polar oxygens and nitrogens. So if these groups weren't here, this molecule may not be soluble in water. However, because it's ionic, it dissolves in water. So now we have a large organic molecule capable of with a lot of conjugation, capable of absorbing visible light, which dissolves in water. And that's what red dye number 40 is. So the next thing I want to talk about is, well, how do we actually measure the absorbance of a sample? And measuring the absorbance of a sample is something you've done before in general chemistry, certainly if you took it here and most likely if you took it anywhere else. And I want to talk a little bit about how this works. Now, I do need to tell you that the spectrophotometers that you're going to use a slightly different method of doing what I'm about to talk about, but overall they functionally do the same job, just with a much lower resolution, and that keeps the cost down, quite frankly, which is important so we can have these spectrophotometers for general chemistry even. So here, what we essentially have is a white light source. So this white light source is putting out the full spectrum of visible light. And if you have a UV vis spectrophotometer, you might have a UV source. You will have a UV source as well. So we have this full spectrum, all the colors, the rainbow of visible light. And it goes through a monochromator. And a monochromator consists of two pieces. One, something that can separate the colors of the light. In this case, we have a prism, which can then separate it into the different colors, like on the Pink Floyd album, or you could use a diffraction grating. If you look at the bottom of a CD, for example, you can see the rainbow. That's more like how a diffraction grating works, and I'm not going to talk about that too much. But ultimately, what you have is the white light being broken up into its component colors. We then put one of those colors through a slit, and essentially that's done by rotating this, and one of those colors goes through a slit. So now we have a single wavelength of light going through our sample. And it's really important to remember that the very first thing you do whenever you use a spectrophotometer is you run a blank. So what happens is you're running your single wavelength of light through your blank. In our case, that's going to be pure water. Water, which is clear, does not absorb visible light. So a certain number of photons are going to hit the detector. And the detector works on the photoelectric effect, which I'm not going to get into in too much detail here, but essentially it converts light into a digital signal that can be read by a computer. So initially, we put a blank here, and we see how many photons of light hit the detector. We then insert a sample, and this sample, in our case, is going to contain red dye number 40, and that red dye number 40 is going to absorb some of these photons of light. So when we look at the photomultiplier detector, essentially what we're having is less photons of light hit the detector. And the instrument reads that as a signal. So if, say, 1,000,000 photons hit the light or hit the detector with the blank in there, and then 500,000 hit the detector when the sample's there, it can convert that into a signal that you're going to read on your instrument, and we'll show you that in just a little while. So that's essentially what this is doing in order to figure out the absorbance. Now, I need to talk briefly about absorbance versus concentration. If you have a dilute red dye 40 solution, there's going to be relatively few red dye molecules between the source of light and the detector. So relatively few photons will be absorbed. If you have a concentrated red dye sample, then a lot of the photons, because there's a lot more red dye molecules, remember the other molecules here are water, which don't absorb this light, so you're going to have a relatively large number of these photons absorbed. Generally speaking, we consider the linear range for absorbance between 0.1 absorbance units and 1 absorbance unit. And you have to make concentrations that fall within that range. That's not going to be super important with the task that you're doing today, but it is in the outline, and it is a good thing to know that that's generally what is considered the linear range. That's not to say you can't be slightly outside of that sometimes, but that's the generally considered the linear range. So this is a little bit hard to decide what order to do things in, but I want to come back again to the visible spectrum that you're actually going to see on a modern spectrophotometer. The microlab is slightly different, and we'll show you that in a few minutes, but there are two types of spectrum, the ideal and the experimental. So remember before we talked about how these transitions, when an electron absorbs a photon, are quantized, meaning that a specific wavelength photon or a specific energy photon can cause a transition of an electron from one state to another. And you would expect to see very specific lines in a spectrum that corresponded to the different transitions. So this would have more transitions, it's a more stable transition. This would have less transitions, it's a less stable transition, so on and so forth. However, what you actually end up seeing is a peak like this. And the reason for this is that the spectrophotometers do not have sufficient resolution to resolve all of these line spectrum. So what we end up seeing, just for practical reasons, is this peak. One thing that's really important to note is Lambda Max. Lambda Max is the wavelength at which you have the maximum absorbance. Remember how we talked about before with the color wheel, red food coloring is going to absorb mostly green light. So green light is around 520 nanometers. So we expect our Lambda Max to be somewhere around 520 nanometers. Red food coloring, not going to absorb red light. It's not really going to absorb blue light, but it's going to strongly absorb green light, its complementary color, because again, white light without green light appears red. So I hope this gives you a basic fundamental idea of some of the theory behind this. Again, UV visible spectrometers are very, very common in chemistry labs, and having a basic understanding of how they work will be very beneficial to you in the future. So the final thing that we want to talk about here is the Beerlambert Law, or commonly referred to as Beer's Law. And you're going to use this during this experiment, and a little bit later on I'm going to show you how practically this can be used with a pen and paper of what we need to solve for. So this is the absorbance equals the molar absorptivity coefficient, which is given for red dye number 40 in your lab manual, B, which is the path length. In our case, this is 1.44 centimeters. It's just how wide the tube that you're going to put your sample in happens to be. And C, the concentration in units of molarity. So we are going to measure the absorbance of an unknown sample of red dye number 40. We're going to use the molar absorptivity of red dye number 40 and the path length, 1.44 centimeters, to determine the concentration. You are then going to make that solution 3 times, starting from solid, as Tim will describe in greater detail coming up right now.
So before we get to the microlab portion of the experiment, I wanted to talk briefly about what you're actually going to be doing in the experiment. Here you can see one of the vials that you'll be using during the experiment to store your solutions when you read them in the spectrophotometer on the microlab. So, that said, what are we going to be doing in this experiment? The first thing we're going to do is we're going to measure the absorbance of the commercial food coloring, of the commercial beverage. Now, there's a couple things to note with measuring the absorbance of the commercial beverage. It's very possible that the absorbance is above the linear range, and it's really important that we have everything must be in the linear range. If you're not in the linear range, then it is possible that your data will not work out the correct way for you, because cutting the concentration in half may or may not have the expected effect on the absorbance if it's not in the linear range. So we have to be inside that linear range, which is 0.1 to 1 absorbance units. So we're going to be checking the measurement of our absorbance of our commercial beverage in the linear range, which we may need to delete to do, or dilute to do, and if we do, we have to work backwards with our math to undo our dilution. So if we dilute it by half and find the concentration to be 1 molar, we need to remember that we diluted it by half, so we have to double it to make it 2 molar. So keep that in mind moving forward. So once we've measured the absorbance of our commercial beverage, we need to find one piece of very important information, and that is the Lambda Max. In order to move forward with the experiment, we need to figure out what wavelength our red dye 40 in the beverage and our solid dye that we'll be diluting in a minute absorbs at. So once we figure that out and we find the concentration of our commercial beverage, we can work on making our solutions to relate to it. So the goal of this experiment is to make 3 solutions with absorbances, and by extension concentrations, that are within ostensibly 1% of the commercial beverage. So we need to do this 3 times. So how are we going to make the solutions? Well, we're going to make the solutions from solid red dye. Now, in the past, you may have worked with some of those dropper bottles of food coloring, and you might have noticed that they're pretty messy. If you get that food coloring on anything, it tends to take the color for a long time, and it's kind of a huge pain to clean. Well, if you thought the liquids were bad, the solids are much, much worse. When you're using red dye 40 as a solid, it's really important that you're super careful with it, because it can be a huge mess and end up all over everything. And then everything will be red for weeks, including the glassware, the spatulas you use, the balances. Everything can turn red for a long time. So you'll want to make sure you're careful when you're using it and that you clean everything very thoroughly afterwards. So when we're trying to make our 3 solutions with absorbances within 1%, it is of course going to be probably unlikely that we can make a solution that is dilute enough to do that. So in order to do that, we're going to need to make a stock solution. So we're going to have to take our stock solution, we're going to make our stock solution, and we're going to try and measure that absorbance. And we want it to be as close as possible to the absorbance that we got in our commercial beverage initially. And if we find that one of 3 things can happen. Either it'll be good, it'll be within 1%, we'll be done with that solution, we can move on to our next one, or we can have one of 2 problems. We can have the absorbance is too high, or the absorbance is too low. Now, one of these problems is much easier to fix than the other. If the absorbance is too high, then that means we have too high a concentration of red 40 food dye molecules in the solution. And in order to lower the concentration, all we need to do is dilute the solution. So we can take our stock solution and dilute it in order to lower the absorbance into the range that we would like it to be in. That's a relatively easy problem to solve. However, we can encounter the harder to solve problem where the absorbance is too low. Because if the absorbance is too low, we can't undilute it, right? We can't remove the volume from the solution. There's just no way to do that. So instead, what we need to do in this case is create a new stock, which would require us, again, starting from solid and making our full solution of red dye 40. So what we want to do is, to recap, measure the Lambda Max and absorbance of our commercial beverage inside the linear range. Then we want to make 3 solutions with absorbances within 1% of the commercial beverage. And remember, this needs to be from solid. We cannot simply measure the same liquid solution poured into the vial 3 times. That doesn't count. We need to start with solid and make 3 separate solutions that we're measuring. And to do that, we're going to need to make stock solution and measure the absorbance until it's within the range within 1% of the commercial beverage. And if the absorbance is too high, we'll need to dilute that stock solution in order to bring the absorbance down to the absorbance of the commercial beverage, or the absorbance can end up too low, and then we'll need to create a new stock solution until it's either at or above the absorbance of the commercial beverage. At that point, we can dilute or write down the concentration and have one of our solutions finished and move on to the second or third solution. In order to make our lives easier while doing this, we can use the Beerlambert Law. If you'll remember, Colin said recently that the Beerlambert Law looks like this, where this is the absorbance, this is the molar absorptivity, this is our path length, which in this case is 1.44 centimeters, which you could either remember or write down from this video, or using the vial in class, just measure the length of the vial using one of the rulers found in the lab, and you'll find that it is 1.44 centimeters, and C is our concentration. So when we make our initial solution using the commercial beverage, we can use this law down here in order to solve for that concentration, which gives us starting points for continuing on with the work. How can we do that? Well, once we have the absorbance, we can rearrange this equation in order to solve for the concentration. So at that point, the equation would look more like this. So now we have the concentration is equal to the absorbance divided by the product of the molar absorptivity and the path length. The absorbance we'll get from the actual spectrophotometer. The path length we can get from measuring or looking right here where we tell you it's 1.44 centimeters. And our third value is the molar absorptivity, which is given in the lab manual or can be looked up online. Either way, you should find the same number, and then you can solve for the concentration. Once you've solved for the concentration, then you'll be able to have a good idea for a starting point for making your solutions. Again, because it's easier to dilute a solution than it is to undilute it, that is, you can't undilute a solution. If you wanted to err on the side of caution, starting at a slightly higher concentration might be in your best interest to then dilute it. However, keep in mind that when you're diluting a solution, you will need to remember how much you're diluting it in order to calculate the new concentration, in order to find the concentrations at which your absorbance matches that of your commercial beverage. Now, I'm going to take some time to show you how the microlab works and how we can use that in order to measure these things. So, now it's time to show you how to use the microlab software in order to measure these absorbances. So what you'll need to do is you'll need to go to the microlab software and open it up. Again, as usual, you need to wait for your reading from flash memory bar to fill, and when it does, the software will open. So, last time I told you that we would be using some of the other functions than just this microlab experiment on the microlab, and we already are. So this time, probably unsurprisingly, we're going to be using the spectrophotometer software. So we're going to go over to the spectrophotometer and double click it. It's going to bring up a small amount of options for you for your which of the spectrophotometer functions you want to do, and then some options. So what we want to start with is the spectrophotometric concentration, because what we're trying to measure right now is concentration and absorbance, so all we need to do is choose the first option, which should be selected for you. Next, we'll need to click OK, and we'll worry about some of those other options later. So there's an interesting hiccup in the microlab software where your blank can be a couple of different things. So I'm going to go ahead and click read blank, and we're going to show you what happens. So if you notice, the spectrophotometer is slowly going through each of the wavelengths. You see here that there's 360 nanometers, 383, 405, and so on and so forth, all the way up to 890 nanometers. Each of these wavelengths is also associated with the color of light that is found at that wavelength. So you can see it goes from the black of the ultraviolet light up through the violet and then continuing onward until you get to the red and eventually the upper region of the reds into the infrared range. So you'll see that as the spectrophotometer is doing its blank, it's very slowly reading each of these columns at each of these wavelengths several times. You can see them, they jump up and then they adjust down, and they slowly come into line with the others over time until they're all reading relatively similar amounts. You'll notice the exceptions are the 555 wavelength and some of the far extreme wavelengths, and that's for the simple fact that those diodes are simply not as strong in the Microlab spectrophotometer. But it's not something that we need to be concerned with. So it's really important that your Microlab takes all of this time to do the blank. There is a possibility when you click read blank that the blank goes very, very quickly and it doesn't go through all of these steps. So I'm going to show you how to recognize and fix that when it happens. You'll notice that as we get to the end of the last wavelength, our progress bar begins to fill a little bit more quickly, and it will reset once until it's ready to go. Now you can see that we have the ability to read samples. What I'm going to do is try to force the program to make an error which will show us a different kind of blank. So I'm going to close that particular function and try again. Again, I'm just going to click on the spectrophotometer and click OK without making any choices. Then I'm going to click read blank and we'll see if I can make it make that error. So you'll notice that this time we're not seeing any bars appear. The progress bar is immediately filling up. If this happens, your blank has not been performed correctly. This will cause errors if you try to run samples at this point. So we need to figure out how to fix it. Generally speaking, if you were to just close the program, reopen it, and read the blank again, it will simply keep doing this, what we call fast blank, repeatedly and never fix itself. So in order to fix that, what we need to do is we need to go back into our spectrophotometer, but this time, instead of clicking OK, we need to click Select Wavelength and Measurement choices. So we're going to click Clear All, and then select one of the wavelengths essentially at random. So I'm going to click the 502 wavelength and click OK. It doesn't matter which wavelength you choose, it just matters that you only choose one wavelength. Once you've chosen it, you'll click OK, and you'll click Read Blank for a third time. This time, we're only reading that single value, so you'll notice that the only wavelength for which we got a peak was the wavelength that I selected in that screen. Now we're ready to force the software to do the long blank. So once it's done this quick single wavelength blank, we can close the software, reopen the spectrophotometer, check our wavelengths and make sure everything is selected again, and then click OK and read our blank. This time when we read our blank, we'll notice that it's going to do a blank similar to the first one, where it's reading each wavelength slowly, doing multiple measurements at each wavelength, and finishing doing the entire thing over an extended period of time. If it does not do this longer blank, then your results will not be accurate, and in some cases they will come out seeming relatively nonsensical. It's very important that you do not simply capitalize on the short blank and be happy that your spectrophotometer did the shorter blank, because it will not work if you do it that way. So for now, I'm just going to let this finish doing its long blank, and we'll pick back up when it's done. So now that our blank is done, we can begin to actually measure the absorbance of some solutions. So here I'm not going to measure the absorbance of any solutions because it doesn't matter what's being absorbed or not. I can still show you how to do it. So the first thing you need to do is, if you look at this graph, you'll see that our x-axis, like I was talking about before, is wavelength in nanometers. But our y-axis is in percent transmittance. Well, generally speaking, we need to be measuring an absorbance because that's what Beer's law calls for, and that's the only way we'll be able to calculate the concentration that we need. So what we need to do is we need to come up to our options up at the top here and choose absorbance kinetics zero order. You'll notice the absorbance for each of these is 0 because it's showing currently the absorbance of our blank. And of course our blank relative to itself is absorbing nothing. It is transmitting all of the light. So in order to add a sample, you need to click add, which will then ask you to give a sample ID, which can be anything you need to do, you want to put in order to identify your sample. So I'll just put in example 1 as our sample name. So I'll leave the concentration or volume at 0 for now and click OK. As soon as you click OK, you'll see that it says reading data and the progress bar begins to fill, and we see that this is the absorbance of nothing in particular, because if you notice, our absorbance values are very, very low. Now, once we get this screen, we can check a few different things. First of all, we can theoretically find our Lambda Max, which is simply the wavelength at which we absorb the most light. So in this case, it's the tallest column, which would be the 700 nanometer wavelength. Again, it's not really absorbing much light because our absorbance is 0.0018 absorbance units, which is roughly 1/50th of the lowest possible absorbance we would like you to have in lab, which is 0.1 absorbance units, because the linear range is considered between 1 and 0.1. So, in order to find our absorbance at that wavelength, you'll notice that currently it shows it's 0. And while the number is relatively small, it is not 0. So in order to make this absorbance reflect the wavelength we want, we need to click on that wavelength. You'll notice that when I clicked on it, all of our numbers changed and it shows the transmittance and absorbance. If I click on any of the other columns, it'll show me the information for that column instead. But for now, we want to stick to our maximum wavelength. So once we run our sample of red 40 and identify our maximum wavelength, we can actually set up the spectrophotometer to save some time in order to not read all the other wavelengths, because if you'll remember, the only wavelength we care about is our Lambda Max. So hypothetically, it will not be the case in your experiment, but hypothetically, let's say that this 700 nanometers was in fact our Lambda Max. In order to use that in all of our future experiments, we would close our software, you don't need to save it, and we'll open a new experiment with the spectrophotometer, and this time we'll select all our wavelengths, clear them all, and select only our Lambda Max, which in this case was 700, so I'll select only the 700 nanometer wavelength, click OK a couple of times, and be ready to read my blank. Now the blank will be quick no matter what, because it's only blanking a single wavelength, and all of our other wavelengths will be automatically zeroed out. Then we can, in fact, read our absorbances, and you'll notice that when I do this, the only column that has any value in it when I switch over to absorbance, the only one that has anything that's changed is in fact our 700 nanometer wavelength, which we can then come over here for the absorbance and use that for the future. But this way you don't need to worry about all the other columns, and only the wavelength of interest will be checked in each of your experiments. So it can save you some time and potential confusion on if you have the wrong column selected. What you do need to do manually, however, is copy all of these values from your absorbances as you change your concentrations, etc. You'll need to copy those values over into Excel manually, as there's no easy way to export this data from Microlab directly to Excel without taking a bunch of unnecessary data as well. So it's easiest just to type in the concentration and the absorbance into Excel directly by hand. So now I briefly want to go over some of the math involved in this experiment. Earlier, Colin told you that you didn't necessarily need an Excel sheet for this experiment, and it's true that you don't. However, having one can help you, as you'll see in a few minutes as I start to fill out this Excel spreadsheet. I can actually have most of this work done ahead of time, so all I need to do when I get to class is measure the absorbance of the commercial beverage, and it'll kind of fill out everything for me as I go. So the first thing you're going to do in your lab is you're going to be measuring the absorbance of your commercial beverage, and you need to determine the concentration. Well, as we covered before, by reorganizing Beer's law to calculate concentration, we need to take the absorbance and divide it by the product of the path length times the molar absorptivity. And in this case, I'm using a molar absorptivity of 2.1 times 10 to the fourth, because this example is relating to sunset yellow and not the red 40 that you're going to use in class. So please don't use my molar absorptivity value for your calculations. But when I hit enter, we'll see that the concentration of that solution is 9.59 times 10 to the negative sixth, and if I go ahead and put my units in, it'll be in molarity. So I need a solution that is 9.59 times 10 to the minus sixth molar. So you'll see here that that number is already filled in, and if I double click on this cell, you can see that I have that this cell is just equal to whatever value gets entered into my concentration cell. So I can have that set up ahead of time so that it automatically updates throughout my sheet. So how do I make this concentration? Well, in order to make that concentration, I need to have a certain mass of sunset yellow. So in order to figure that out, I need to turn my concentration into a number in grams. So if I have a molarity value and I multiply it by a volume in liters, in this case 250 milliliters or 0.25 liters, because that's the volume that I've chosen to use for this example, it'll convert it into moles. Once it's converted to moles, I need to convert it into grams by multiplying by the molar mass. So when I do that, I see that the number is 1.08 times 10 to the negative 3. E in Excel and in other places means times 10 to the. It's a shorthand to save space in the limited cells. I see that I need 1.808 times 10 to the minus 3 grams of sunset yellow, which is about 1 milligram of solid. Now, the balances in the labs are capable of displaying numbers with that kind of decimal points. However, it's not a reliable amount to measure. So in order to have a more reliable amount to measure, we can go with a small number that we know we can reliably measure on the balances. In this case, we want to target around 50 milligrams because that's a balance or a mass that we can fairly reliably and repeatably measure on the balances. So in this case, we've hypothetically measured out some grams of sunset yellow, and as you can see here, it ended up being 0.0528 grams or 52.8 milligrams. So now I want to calculate the concentration of this stock solution. So I want to take these grams and convert them into moles, and to convert it into moles, I need to divide by the molar mass. Once I've converted into moles, I need to divide by my volume in liters, which will give me a molarity. In this case, about 4.67 times 10 to the negative fourth molar. Or in this case, you can see we have a whole bunch of sig figs here that Excel is giving us. So now we have a stock solution with a concentration of 4.67 times 10 to the minus 4 molar. However, the solution we want to generate is 9.59 times 10 to the minus 6 molar. So how do we do that? Well, we use our dilution equation, C1V1 equals C2V2. So we need to take the product of our target molarity and the volume of that solution we want to make, in this case 25 milliliters because that's plenty to make our measurement, and we want to divide it by the concentration of our initial solution, in this case our stock solution. This will tell us the milliliters, or rather the liters of stock solution we should use to make this solution. In this case it is 0.000513 liters, which when converted to a more usable number is 0.513 milliliters, which we can measure using our micropipetters that we used last week in order to make our solutions. So theoretically this will work perfectly and our absorbance will come out to be exactly 0.29 and we'll be happy and we can go on our merry way. However, in reality that doesn't always happen. So what we need to do is, what I'm going to do is show you a couple of different ways to account for your math not working. Or rather your absorbance being higher or lower than expected. So down here you'll see I have the heading, the absorbance is too low. So let's say theoretically that our desired absorbance was the same 0.29 that we talked about before, and our recorded absorbance was 0.272. So last time when we made our solution, we used this volume of our stock. Well, now we need to use a new volume of our stock in order to make our solution, and we want it to be a higher number. Well, what number will we use? Well, to figure that out, we want to set up a ratio, because the ratio of the dilution is the same as the ratio between the absorbances, because we're on the linear range. So, in order to set that up, what we want to do is we want to take the absorbance that we measured using... Sorry, we need to start with a parentheses. We want to take the absorbance that we measured, and we want to, or the absorbance that we want, our target absorbance, which is 0.29, and we want to multiply that by the volume that we used. After we've done that, we want to close our parentheses and divide by the recorded absorbance. So what this essentially does is divides our ratio of our absorbances and multiplies it by the volume of the stock we used, and that will give us a new volume of stock with a ratio equal to the ratio between our absorbances, which works because the absorbance values are linear with respect to concentration. So when we hit enter, we see that instead of using 0.513 milliliters of our stock solution, we want to instead try to use 0.547 milliliters of our stock solution and generate a solution with a total volume of 25 milliliters, just like last time, and check that absorbance. And we would expect that absorbance to be much closer to the 0.29 that we like. Alternatively, if the recorded absorbance is too high, we don't need to make a new solution. Instead, we can simply dilute our current solution. And again, we can use the ratio between our recorded and our desired absorbances to figure out what ratio to use. So, again, we want to make a 25 milliliter solution, so we need to figure out how much we need to dilute this solution by. So, when we look at this value, we just need to do a little bit of math to find what we want. So in order to do this, we need to calculate the concentration of our solution as what we want it to be, because the concentration we targeted for, the 9.59 times 10 to the minus 6th, ended up being too high in this scenario. So to do that, the first thing we need to do is we need to find the ratio of these and find out what our new concentrations should be. So our concentration of the new attempt solution in molarity will be equal to the ratio of our recorded absorbance and our desired absorbance, but it's got to be the ratio of our desired to our recorded, because we want the number to get smaller, so we need the value to be lower than 1, and we're going to multiply that by the concentration of the solution we used the first time. So that'll tell us what the concentration we want to use for our new solution is. Now it's simply a matter of using C1V1 equals C2V2 to determine how much of the first solution we should use. So we want to take the concentration of our new solution times its volume and divide by the concentration of our initial solution to find that we should use a 0.0232 milliliters of that liters of that solution, or 23.2 liters. So we want to take 23.2 liters milliliters of our first solution and dilute that down to 25 milliliters using 1.8 milliliters of water in order to create a new solution which should hopefully have an absorbance closer to the 0.29.